Hello class, these are the mutation notes for Unit 5 protein synthesis. Let's start with an example like sickle cell disease. Sickle cell disease is where you have red blood cells that instead of being the nice inner tube shape that can flow really easily through your cells, they are instead shaped like a sickle. A sickle is this farming tool used for cutting grasses and wheats and grains. So this sickle shape actually prevents it from flowing very easily, and it can cause clogs in your blood vessels, which can prevent the transport of oxygen, sugars, antibodies, anything that your body might need. But this is caused by mutation. So mutation is any change in DNA, and it usually happens during replication. It can happen other times, but the cells are often able to proofread or fix this, but sometimes they don't, and it can get passed on or cause other damages. Some mutations are beneficial, but most of them are probably going to be harmful. So you're going to need to know a couple of things. You're going to need to know what causes mutations, what are the main types of mutations, and what are their effects and some examples of them. Most of your focus should go on these two. So we've already talked about carcinogens when we talked about cancer. So carcinogens were those things that can cause cancer, that can damage your DNA and lead to some change in your cell cycle. Carcinogens are a specific type of mutagen, but a mutagen can be anything that changes DNA, whether it causes cancer or not. So a lot of examples could be aerosols, pesticides, tar from smoking, lead paint. Viruses can change your DNA. Radiation. So UV rays are a very common mutagen. Now, if mutations happen in your body cells, they can damage you, but they're not going to necessarily get passed on to your offspring. If you have lung cancer, that's just going to stay in your lungs. It's not going to get passed on to your offspring. However, if mutations happen in your sex cells, then they can get passed down. So if you get radiation that changes some of the DNA in your sperm or eggs, then that can affect your offspring. So why does it matter? Why does it have to change the DNA? Well, the DNA codes for the proteins. If you change the DNA, you can change the amino acids that make up the proteins, and the proteins make the traits. Those blood cells were made up of proteins, and the wrong proteins made them have the wrong shape. By changing the DNA, we can do that a couple of different ways. We can substitute the individual bases. We can insert bases, or we can even remove bases. But if I'm just adding or removing or changing bases, these are just called point mutations. They can have pretty much all the ranges of effects. So if it's a substitution, if I just change one base, if I just change UUU to UUG, well, UUU codes for phenylalanine. I can see that on the codon chart here. If it was changed to UUG through a mutation, it's going to code for leucine. That's a different amino acid, and it's going to lead to a different protein. That protein could be better, but it's probably going to be worse. Now, if I changed UUU to UUC instead, well, if I look over here, UUU is phenylalanine. UUC is also phenylalanine. So this mutation is actually not going to do anything. It's going to be the same amino acid. It's going to be the same protein. If I change the amino acids, this is called a missense. It's not making sense. It's a different kind of mutation. It's a different kind of protein. A silent mutation is not doing anything. It's the same amino acid. It goes unnoticed. There are many known mutations that cause things that are just changes in one base, one point mutation. For instance, sickle cell disease that we were talking about earlier with the blood is just one change in a base. Instead of being GUG, or instead of being GAG, I turn it into GUG. So it's a missense. It's a different amino acid, and it makes different proteins, and it messes up the whole structure. Now, if I add or remove a base, it can change the way that I read the bases. That's called a frame shift mutation. So let's just take a look at the sense. The normal sense is the fat cat ate the rat. This is a good example because all the letters are three, or all the words have three letters. Just like RNA gets read in groups of 3. That's a codon. So if I change this C here, then everything gets moved up. So the next group of 3 instead of cat is now ATA. So this A from the other one got moved up to this one. And everything after the C is now different. So instead of the words being ate the rat, it's now atta, tet, her, at. And it's totally different. Doesn't even make sense. The same goes with insertion. If I add a base in here, if I add in a nucleotide, I'm going to shift the frame in which I read. The groups of 3s will now change, and it can cause a lot of damage because all of the amino acids after that are different, and I have a very different protein. So in terms of the amino acids, so I have this T and C here. CAT codes for histine, CAT again for histine. If I was to add in a nucleotide in between the T and the C, then I'm going to change the groups. Instead of reading CAT, I'm reading ACA, and that's going to make a tRNA drop off a threonine instead of a histine. So again, all these amino acids are going to be different. The whole protein is going to be different. And they don't have to just be with bases. They can be whole portions of chromosomes. They can be whole chromosomes. They can be portions of chromosomes. I can duplicate a section of this chromosome. So this section of the chromosome probably has tens or hundreds of genes on it. And if I duplicate it, that's going to really mess up the way that my body's reading those genes. We could also invert it. We could also lose whole portions of the chromosome or translocate them. Some cancers like leukemia are caused by these translocations. Adding whole chromosomes would be like Down syndrome that we talked about before. But these are huge portions of genes that can cause a lot more changes. Some other mutation examples are, other than the sickle cell disease, could be like blue eyes. Blue eyes was originally thought to have been caused by a mutation in the pigment that makes your eye. Lactose intolerance is caused by a different protein that's supposed to make the lactase enzyme that breaks down sugars in milk. Red hair is a change in the protein that makes the pigment of your hair. Albinism is caused by a change in the pigment that makes your skin. These are all protein-based. And of course, resistance to a lot of different diseases like HIV and the bubonic plague are thought to have been caused by mutations. Now the graph over here is reading 2 things. It shows us frequency of mutations and it shows us fitness. Now fitness is a measure of how well adapted an organism is. So if it's less adapted, it's going to be a lower fitness. If it's more adaptive, it's going to be a higher fitness. So the frequency of mutations can change the fitness. And as you can see, the higher the frequencies are, the higher frequencies anyway, highest is down here at 0. It's really harmful. If it's less than 1, that's harmful. If it's equal to 1, that's neutral. If it's greater than 1, that's beneficial. Most of the frequency of mutations is below 1. All of these are harmful mutations that happened. At 1, these are mutations that really didn't do anything, and above 1 are beneficial mutations. So as you can see, most mutations aren't really helping organisms because we have a very low amount up here. So to review, mutations are caused by mutagens. This can be anything from X-rays to cigarette smoke, other types of radiation, chemicals in the air. A lot of different things can be mutagens. The different types of mutations are going to be, we have point mutations, which can be substitution of different bases or addition and deletion of bases. Substitutions can be missense, where they read incorrectly, or nonsense, where they don't do anything. Sorry, that's actually incorrect. Shouldn't be missense. It's the one that doesn't do anything. It's the silent mutation. So instead, it would be silent. There's nothing that's happening. Addition and deletion cause those frame shift mutations where they change huge portions of the DNA. Chromosomal mutations can change whole sections of the chromosome, and most mutations are going to be harmful. Some of them are not really doing anything. Some of them are actually beneficial. Well, that was mutations. I hope that was helpful.